Hello, my name is Megan Wiesman, and I will be discussing the role of the pancreas and liver in regulating digestion. The pancreas contains both types of secretory epithelium, both exocrine and endocrine. Exocrine secretions include digestive enzymes and sodium bicarbonate. These secretions come from lobules called acini. These lobules of the pancreas are very similar to the ones that you find in the salivary glands. Most pancreatic enzymes are secreted as zymogens. Zymogens are activated in the intestine after being in contact with the brush border enzyme enteropeptidase. Enteropeptidase will convert the pancreatic zymogen trypsinogen to its active form, trypsin. Trypsin will then convert other zymogens into their active forms. These enzymes include chymotrypsin, which is used in protein digestion, carboxypeptidase, which works on the C-terminal end of proteins and peptides, colipase, a cofactor for pancreatic lipase, and phospholipase, which breaks down fatty acids. The signal for these enzyme secretions include the enteric nervous system, which is stimulated by the distension of the duodenum, neural signals like the parasympathetic stimulation via the vagus nerve, and CCK, a hormone made in the small intestine in response to fatty acids entering the duodenum. Bicarbonate secretions function to neutralize stomach acid. Stomach acid is a very strong acid with a pH of about 2. Without this neutralization, the small intestine epithelium would become damaged. Synthesis of bicarbonate requires high levels of carbonic anhydrase. Bicarbonate is produced from a reaction of carbon dioxide and water and then secreted on the apical surface by a chloride bicarbonate exchanger. The extra hydrogen ion leaves the basolateral membrane via a sodium-hydrogen exchanger. The bicarbonate production is stimulated by secretin also from the small intestine or the duodenum. Secretin will increase the production and release of the bicarbonate. The pancreatic endocrine cells are clustered into islets. Most of these cells are beta cells which secrete insulin and amylin, but some are alpha cells which secrete glucagon. Insulin is secreted in response to high levels of glucose in the extracellular fluid, which occur in instances like after a large meal. This large meal or these high levels of glucose trigger the exocytosis of insulin containing secretory vesicles. Insulin will then travel through the bloodstream to the liver, muscle, and fat cells and signal to these cells to take up and store glucose, which decreases the blood glucose levels. Amylin, which is also secreted by the beta cells, helps to control appetite and the amount of food leaving the stomach. Amylin slows gastric emptying and reduces gastric secretions. Additionally, amylin promotes a feeling of fullness and decreases food intake. Glucagon, which is secreted by alpha cells, acts in opposition to insulin. If blood glucose levels fall too low, glucagon will be secreted, which signals to the liver to break down glycogen and release glucose into the bloodstream. The liver functions to aid in digestion as well as in some waste elimination. Bile, which is secreted from the liver, contains acids and salts to aid the breakdown of fats during digestion. But there are also some liver waste products like bilirubin, which is created when old red blood cells are recycled and hemoglobin is broken down. This is found in bile and will be removed by the body in the form of feces. The liver secretions are controlled by two hormones, CCK and secretin. CCK is released in response to fat entering the duodenum. When released, CCK will stimulate the gallbladder to contract. This forces bile into the common bile duct and then into the duodenum. Secretin increases in response to acid in the duodenum. The action of secretion in the bile duct is similar to its action in the pancreatic ducts. It signals to the duct cells to secrete bicarbonate and water. This secretion will increase the volume and flow of bile from the common bile duct. Bile is composed of many things: bile salts, bile pigments, phospholipids, electrolytes, cholesterol, and triglycerides. but of greatest importance to digestion are the bile salts and phospholipids. Both phospholipids and bile salts are amphipathic, which allows them to act as emulsifying agents. The molecules have both hydrophobic and hydrophilic sections. The hydrophobic regions will surround the large fat droplets in the duodenum. This leaves the hydrophilic sections to interact with the more watery chyme. The phospholipids help to separate the large droplets into smaller ones by this surrounding action and increases the available surface area. This increased surface area allows for lipase to bind more quickly and break down the fats. This is very important because if these emulsifying agents weren't present, the droplets of fat would be too large for lipase to effectively bind to and break down. Therefore, little fat would be absorbed from the small intestine. Dietary fats are necessary and used for a variety of functions throughout our body. These functions include organ insulation and protection, warmth, energy, and production of some hormones. In conclusion, the body needs to break down the complex molecules that we ingest into small, more easily absorbed molecules. Enzymes are very necessary to allow these reactions to occur in a usable time frame. Pancreatic enzymes are important in the digestion of fatty acids, proteins, and help lipase to function. Bile, which is formed in the liver and stored in the gallbladder, is used to digest fats by increasing the surface area of the substrate. Both pancreas and liver secretions are controlled by CCK and secretin from the duodenum. However, the pancreas can also be stimulated by the parasympathetic nervous system via cranial nerve 10, the vagus nerve, and the enteric nervous system by the distension of the duodenum.